Hey guys, we've been talking about passive transport, diffusion, osmosis, um, how to figure out which way the water's going to flow in solutions. Um, and the next we're going to talk about is active transport. All of these up here did not require the cell's energy to have substances move into or out of the cell. But when we get into active transport, it does require the cell's energy. So I'm going to open up this link here. So in contrast to facilitated diffusion, which does not require energy and carries molecules or ions down a concentration gradient, active transport pumps molecules and ions against a concentration gradient. Sometimes an organism needs to transport something against a concentration gradient. That means, so normally in natural settings, things move from high concentrations to low concentrations. Here it may be a low concentration and it needs more of something else. So it's going to make it move into a higher concentration. It's going to make it move opposite of what normally happens. And this is done through active transport, which uses energy in the form of a molecule called ATP. So energy for cells is this molecule called ATP. It's pronounced adenosine triphosphate, and it's produced by cellular respiration. And that is in the mitochondria. That's where the oxygen that you breathe in gets into your bloodstream. It goes to your cells, and the mitochondria uses the oxygen. That's why it's called respiration. Your cells don't have lungs. Remember, I went that whole spiel about the mitochondria uses food and oxygen to break down and release energy. This is what they're talking about here. And that energy carrier molecule is an ATP molecule. So active transport is the energy requiring process of pumping molecules and ions across membranes or uphill against a concentration gradient. The active transport of small molecules or ions across the cell membrane is generally carried out by transport proteins that are found in the membrane. Larger molecules, such as starch, can also be actively transported across the cell membrane by a process called endocytosis and exocytosis. Okay, so that means that endocytosis and exocytosis are also forms of active transport, and they require the cell to expend energy to do so. And you may think, well, why is that a big deal? Well, when you think about a cell, something microscopic, and it has to have something move, that expenditure of energy for that tiny of an object is a lot. Okay, so we're going to watch this video, and then we'll help you fill out your study guide.
Many substances move in and out of cells. In order to get substances into or out of the cell, they must get through the cell membrane. Let's take a closer look at the cell membrane. The cell membrane consists of a phospholipid bilayer. Phospholipids consist of a phosphate head that loves water, or hydrophilic, and a lipid tail that hates water, or hydrophobic. The phospholipids arrange themselves into a bilayer with the head pointing outward and the tails pointing inward. Lipid-soluble substances can pass through the phospholipid bilayer. Some examples of lipid-soluble substances include oxygen and carbon dioxide. Steroids are another example. We can say these substances move by diffusion. In diffusion, substances move from higher to lower concentration until reaching equilibrium. Here is an example of diffusion. A drop of dye in water will move from where there is more dye, an area of higher concentration, to where there is less dye, an area of lower concentration. The molecules will eventually evenly distribute throughout the solution. Non-lipid soluble substances cannot pass through the membrane, so they need a special doorway called a protein channel. In this example, sodium moves into a cell through a protein channel. Other examples of substances that move through protein channels include glucose, chloride, and potassium. We can say that these substances move by facilitated diffusion. In facilitated diffusion, substances move from higher to lower concentration until reaching equilibrium, but through a protein channel. Substances can also move against gradients. Here we have more sodium outside of the cell than in. If sodium moved by facilitated diffusion, it would move into the cell. However, energy can be used to move sodium against its gradient, or outside of the cell. Substances can move against their gradients using active transport proteins. These proteins use ATP for energy. An example of an active transport protein is the sodium-potassium pump. The sodium-potassium pump moves 3 sodium outside of the cell and 2 potassium inside of the cell for every molecule of ATP used. The sodium-potassium pump works to maintain concentration gradients. In osmosis, water moves through a semi-permeable membrane, which is a membrane that allows water through, but not solute. Water will move toward an area of higher solute concentration. If a beaker is separated into 2 compartments by a semi-permeable membrane, and each compartment has a different solute concentration, water will move toward the area of higher solute concentration. Tonicity or concentration can be described in terms of isotonic, hypotonic, and hypertonic solutions. Isotonic solutions have the same concentration as body fluids. Hypotonic solutions are less concentrated than body fluids. Hypertonic solutions are more concentrated than body fluids. In this experiment, a red blood cell is dropped into a hypotonic solution. Water will flow into the red blood cell because the cell contains a higher solute concentration. The cell will swell and burst. Likewise, if a red blood cell is dropped into a hypertonic solution, water will flow out of the cell, causing it to shrink or crenate. Water again flows toward the higher concentration of solute, which this time is outside of the cell. Other methods of moving substances in and out of cells include exocytosis and endocytosis. In exocytosis, vesicles inside of the cell adhere to the cell membrane and release substances. Neurotransmitters are released this way. In endocytosis, the cell membrane enfolds around substances to bring them into the cell. There are different kinds of endocytosis. In phagocytosis, the cell engulfs material such as debris or bacteria. In pinocytosis, the membrane enfolds around extracellular fluid to bring it into the cell. In receptor-mediated endocytosis, substances attach to membrane receptors that signal the cell membrane to enfold around the substance. We hope you have learned something about cell transport, and see you next time.
Okay, so this video touched on a few things that's on your study guide. So let's go back over here. So with active transport, you need to know that substances are moved against a concentration gradient, which means substances move from low concentrations to high concentrations. From low concentrations to high concentrations with the use of the cell's energy. Okay. Now, they mentioned exocytosis, endocytosis, phagocytosis, and pinocytosis. So these are vesicles. So that's why under here it's called vesicle transport. So exocytosis, what can happen here is a vesicle can bond with the cell membrane and push substances out of the cell. Okay. And I wish I had a way to draw here. Let me see if I can do. I don't think I can draw with this one. Okay. Anyway, that was the diagram where you had the cell and there was a little vesicle inside. It bonded with the cell membrane and it pushed the substance out of the cell. This one here is where the membrane can wrap around a substance in the environment, encapsulating it and moving it into the cell. Now, phagocytosis is a form of endocytosis, and it's like when it engulfs a solid. So phagocytosis is a form of endocytosis, and solids or particulates are engulfed, and solids are engulfed by the cell. Pinocytosis is another form of endocytosis, and fluids are taken into the cell. Okay. All right. And cellular transport is important to cells maintaining a chemical balance or to maintain homeostasis. So here you can go down, write down the 3 types of passive transport, again, as a review. Okay, so you've seen me write this, so I'm going to back out of this because it may be changing the study guide, and I don't want to change other people's stuff, so back out of this, and that way you can fill this in on your own, but you can do a split screen if you need help with some of these answers, but at least you can hear them or you can write them on a piece of paper off the video, and then you can transpose them back onto your document. So I'm going to go up here for just a moment, back out of this. Remember, you can watch the video and get the answer. So I'm going to look at vesicle transport here and see if they have another video here. Okay. So here they're just talking about exocytosis. You'll have a vacuole inside of the cell. The vacuole moves to the cell membrane. It opens up and pushes stuff out. Endocytosis, the cell membrane starts to wrap around substances and pull them into the cell. Because it's a particle, that is phagocytosis. If it was a liquid, it would be endocytosis. So phagocytosis, cellular eating, dissolved materials enter the cell, engulf solid. Penocytosis, it refers to as cellular drinking. Plasma membrane folds inward to form a channel allowing dissolved substances to enter the cell, such as fluids. Okay. All right.
This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane. They leave the cell instead by a process known as exocytosis. After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus. Their membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As membranes merge again, the contents leave the cell without actually crossing the plasma membrane. This process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis.
Okay. So hopefully this will help explain a little bit of what these documents are talking about and the difference between active transport and passive transport, which we will have a test over this probably on Thursday. So you need to know your types of passive transport. You need to know the difference between hypertonic, hypotonic, isotonic. You need to know active transport requires cell's energy. You need to know the difference between exocytosis, endocytosis, and then the types of endocytosis. Okay.